Dr. Ennio Tasciotti, coming from Houston, and he's an associate professor of nanomedicine at the Institute for Academic Medicine, the Houston Methodist Hospital Research Institute. He's a director, and he's going to talk about Leukolike technology to avoid nanoparticle recognition by the immune system. That's great.
Thank you so much. All right, well, my presentation goes up. I'm going to anticipate that compared to the experts that presented before, I'm not an expert in immunology, and what I'm going to present you is a little bit of a different approach that we took on nanoparticles to overcome some of the limitations that over the years we realized kept coming back. And some of these limitations are linked to how the biology of our body, and the fact that our body does a very good job at protecting. Thank you so much. Does a very good job at protecting us from the invasion of foreign species, foreign objects. And it does so by using biological barriers, developing biological barriers. And these are physical or biological obstacles that prevent bacteria, viruses to just roam free in the body. And the problem is that these same biological barriers also make the life particularly difficult for nanoparticles. And that's because mainly two, these are the two I'm going to focus on, the mononuclear phagocytic system, also known as reticuloendothelial system, in general macrophages and immune cells, and the endothelial barriers deeply limit the efficacy of nanoparticles. Nanoparticles are typically injected in the bloodstream, they circulate in the bloodstream, and yet very rarely the problem is in the bloodstream. Typically the problem is around blood vessels, and endothelial barriers really create a limitation of the amount of nanoparticles that can reach the problem. Similar, the mononuclear phagocytic system is constantly on the lookout for nanoparticles, and they typically remove them from circulation very quickly. Now, the way the industry, the field, has addressed this problem is from the standpoint of chemistry. So they've used a lot of chemical modification of the surface of these particles, typically with this brushing technique. So using highly hydrophobic molecules that prevent the opsonization of particles, or actually by intentionally opsonizing particles, so binding to the surface of particles some of these proteins. Similar approach for targeting, just using antibodies or ligands. And these are kind of the typical approach that we've seen for the past 20 years. I have a background in molecular biology, molecular medicine. I'm not an engineer, I'm not a chemist. I really didn't know exactly how to contribute to the field. But if going back to biomimicry and bioinspired solutions, try to find inspiration in how nature has solved some of those problems and develop technologies that borrow those solutions from nature. And this is something that it's maybe can be described by this technology, which is the gecko feet, imitates the gecko feet. Geckos can climb on walls and mirrors because of the presence of these nanostructures on their feet. The Department of Defense developed these special gloves with nanofibers that allow soldiers to climb on surfaces, and it's all about matching composition, structure, and function. And I'm gonna try to convince you how, with the platform that we developed, we try to imitate the composition, structure, and function of a biological structure to solve some of the problems that we have in nature. Now, the way I see the relationship between macrophages and nanoparticles is pretty much the relationship that exists between a predator and a prey. Macrophages being the predator and nanoparticles being the prey. Now, when you look in nature and you look at how the animal kingdom, in this particular case insects, have dealt with it, now you have a lot of predators and a lot of preys, and very often preys try to mimic the aspect of predators just to hide in plain sight. And if you take this very simple deceitfully simple concept and you apply it to technology, what we propose is, okay, so we have macrophages that can recognize nanoparticles. What if we extract the macrophages from our patient, we develop a protocol, a way to strip the membrane of these cells, and then we mount the membrane of these cells around nanoparticles. So we pretty much dress nanoparticles as macrophages. And with the hypothesis that this would allow us to do three things that macrophages and leukocytes in general do very well. They obviously, they're not recognized by other macrophages because they're like one of their kind. They can target inflamed endothelia very efficiently. That's what they're programmed to do. And they can also penetrate deeper in tissues and ideally penetrate and integrate themselves with the cancer mass. And so this was the idea. And it took us like 3-4 years, but we published this paper in 2015, 2013, in Nature Nanotechnology that we called Open Sesame, this technology. So these particles, dressed like cells, can now open all of those biological barriers, all those gates that I mentioned before. We obviously did a lot of chemistry to make sure that the lipid bilayer with all the proteins integrated were well-oriented, so they maintain the right orientation. That's very important. And you see that now particles go from something like this, a porous silicon, to something that looks a little bit more like a cell. But besides that, what is important is that macrophages cannot really, can't tell the difference. And when you do an uptake assay, so you feed to these yellow macrophages red uncoated particles and green coated, so leukolike particles, you see that over time the macrophages invariably eat the uncoated particles, but they cannot spare the coated particles. And this is true in a static system, but it's also true in all of these videos that I'm going to show to you. So this is what happens when a population of macrophages is exposed to particles, like naked silicon particles. They pretty much eat them right away, and you can see it much better here. You see this particle now will be recognized, sensed, and internalized by these macrophages. And all the other particles in this field of view, they're all internalized by macrophages pretty effectively. In the case of our particles instead, not only macrophages were not even activated, so they were very quiet, quiescent, they did not move, even if particles were floating and flowing on their surface. And you see also in this other experiment that they were maybe associated with the membrane, but they were never internalized. And this gave us some sort of kind of hope that we were onto the right path. But at that point, the technology was still a little complex. We had a silicon core coated with patches of membrane. It wasn't very translatable. We do a lot of translational work. And so we kind of went to a simplified system, which took other 3 years. And we just published that in Nature Material. This is not the cover. This is the image that I sent to them. I hope it's going to be picked. And in this case, what we did is we isolate the membranes, but rather than using them as coating, we recircularize them. So we make vesicles now that contain all the proteins of the leukocyte, but they're like around 120 nanometer in size. They're pretty regular, as you can see. They have all the right domains on the surface. But most importantly, they contain the whole spectrum of proteins that are associated with a membrane, with the membrane of a leukocyte. So the idea is, all right, well, we have all sorts of proteins, integral protein, peripheral protein, lipid anchor protein, cytoskeleton protein. Somehow we carry along the entire complexity of the molecular machinery that is associated with a leukocyte. And this makes these vesicles now, these 100 nanometer proteolipid vesicles, very similar to something the cell produces regularly, like exosomes, ectosomes, or extracellular vesicles. So now we have created a class of nanoparticles that look and are composed exactly of the same components of something the cells make on a regular basis. And this is like a cartoon that I got from a Nature review on exosomes, and it's kind of color-coded with all the different classes of proteins present. And you see that this is what we make, and I respect the color coding, but the sense is that we have the same type of proteins that have the same function. And that's to go back to that original slide where I said that if you match structure, composition, and function, you go a great deal of, you get a great deal of advantages in terms of biomimic. And so now we have vesicles that have all of these type of function. They have proteins that do transport, proteins that do signaling, proteins that take care of immunity, the whole complexity. And the important thing, though, is what happens in vivo. And so we moved from a lot of live microscopy to intravital microscopy, where our objective, our specimen is no longer a slide, but it's an animal. It's a live animal that we anesthetize. We open these little windows on top of different organs that we want to look at. And then we follow over time what happens. And I don't know if we can have lower lights, because otherwise we're probably not going to see the fluorescence. But what we do here is we follow what happens in the liver. And these are the liver sinusoids in green. In blue, you have macrophages, or Kupffer cells. And you see that a normal libosome, if they get internalized by these, trapped by these cells pretty quickly, while in the case of the biomimetic, the leucosome, the proteolipid vesicles that I just showed to you, there are very minimal uptake. So this was another confirmation that our system could truly escape immune recognition. And you see this, that there is just a minimal uptake in red of these biomimetic particles, while there is a major uptake right away upon injection of the other ones. So that results in a beautiful thing, because now you have way more particles circulating in the animal that you had in the case of the control particles, because those are all stuck in liver and spleen. And so this was, of course, we also quantified that, and we had a beautiful increase in the circulation time. But what happens to these particles? Now we have these particles that are circulating along the blood system, and we have around 10,000 miles of blood vessels between capillaries. It's an incredible highway system that our body has. So how do you find the right spot? How do these particles locate the area where the payload has to be released? And another biological event comes in our help, and that is inflammation. Inflammation is the common denominator of a lot of pathological problems in the body. It's kind of a common denominator of all of them, but it's in particularly important also in cancer. It's one of the 7 big hallmarks of cancer. And we typically use already inflammation to our advantage in nanomedicine, because inflammation and leaky vasculature, it's the reason why a lot of nanoparticles accumulate. There is an increased permeability, and this is the EPR effect. But we wanted to extend that, and we wanted to kind of take advantage of this part of the game, where cells now arrive to the inflamed vessels, recognize the inflamed vasculature, and can adhere to the endothelial layer and penetrate the tissues. And we did that with our system, with the hypothesis that the normal vesicles would never stick to a normal endothelium, because it doesn't have the receptors that bind the molecules that we attached on their surface. And you see, this is it's an in vitro flow experiment. So we created these monolayers of endothelial cells. We apply flow on top, and we flow particles. And so you see that you have a few liposomes that, again, get internalized, way more leucosomes when the endothelium is inflamed. And also, if you inhibit, and so if you use antibodies against LFA1, CD45, MEC1, all the other proteins that we introduce on these proteolipid vesicles, now you reduce the uptake greatly. And this is also true in vivo, obviously. So we went back with intravital microscopy and we checked again if we could accumulate our red-labeled leukosomes in inflamed vessels compared to not inflamed vessels. And this was a very simple model of ear inflammation so that we could have the contralateral ear as a control. But we did that also in cancer. So what you see here, it's liposomes being injected in a syngeneic, in a mouse carrying a tumor. This is a breast cancer lesion. Maybe you gotta see it again. And you see that the liposomes find a little bit of accumulation through the EPR, but when we move the animal to inject the leucosome, you see that there is an incredible accumulation of these nanoparticles in the context of the breast vasculature. And we took the time also to measure this accumulation with these fancy systems. And so if you now focus along these two lines, you see now that along this capillary there is the red signal that starts to pile up exactly along the endothelium. You'll see this very thin red signal. And that was the sign that our leukosomes could adhere stably on the endothelial barrier, which is one of the steps necessary. And finally, the other step that we wanted to observe was extravasation. So now again in green we have our capillaries, and you see the nanoparticles progressively coming out of the vasculature and penetrating deeper in the tissue. So this was one of the first time, or the first time, that we could observe in a live animal the process of extravasation in theory, in the course of happening. And this is like all the quantification that we did. We've used, I have 2 more slides, so I'm going to wrap quickly. We've obviously used these particles as a drug delivery system, even if we compared them with Doxil, which is the obvious standard of care. And the most important thing to me is not just the fact that the leukosomes carrying doxorubicin could reduce the tumor burden, but the fact that the empty leukosomes could also have a therapeutic effect of sorts. So now what I really like to do, and what we're very interested to do in the lab, is to understand how come that this nanoparticle, even without any payload, has the ability to somehow trigger a response in the body and elicit some beneficial effect against cancer. And when we started to look at all the immunological aspects, inflammation, chronic inflammation, systemic, local, we couldn't really find anything bad about these particles, even if, and we did all of this because the reviewers of the paper were very adamant to understand if we were eliciting some autoimmunity, because we were exposing some epitope that wasn't normally there. But we did all the experiment and we couldn't see any greater increase in IgM, IgG. Now you see these are a little bit taller, these bars, but we're talking 0.1%, 0.2%. So this is really minimal increase in terms of auto-antibodies. And when you look at all the different organs over time, you see that there is a perfect histology outlook that both macroscopically and histologically there is no evidence. And so the take-home message is that it's possible to create biomimetic systems inspired to the biology of particular classes of cells that have unique capabilities that are very good for drug delivery, and that these particles can escape the reticular endothelial system, can target inflamed endothelia, can extravasate out of the blood vessels, penetrate deeper in the tumor, and provide some sort of therapeutic effect. This is mainly the work of Alessandro Parodi, a very talented postdoc who's now assistant professor at the University of Chicago in Illinois. Roberto Molinaro, that's the first author of the Nature Material paper. Claudia Corbo did all the proteomic. I have a big team that does imaging and all the people that did the immunology studies. And I really want to acknowledge them for all their work. Thank you.
Thank you very much for the wonderful presentation. This is amazing technology. We have time for one quick question, and I suggest to, if you have more questions, that to address them to the speaker after this session.
Oh, yes. I'll be around.
Right.
Hi, that was really interesting talk. The images are absolutely incredible. My question, I suppose, is a little bit technical. I mean, I've seen people in the pharmacology space, they've used that sort of cell disruption technology before to create vesicles to look at drug transporter activity. And 9 times out of 10, what they use it for is to look at the ABC transporters that you mentioned, the efflux transporters, so they'll create a vesicle from the cell that they're interested in so that the transporter then is inverted and it accumulates within the vesicle. So I noticed that you mentioned on the same slide where you talked about that reconstituted membrane, there was a graph for orientation. How do you guarantee that the proteins, that when the membrane sort of reforms, if you like, into the vesicle, that the proteins are still sort of pointing out, if you like?
So we do it 2 ways. There is a gross way to do it, and it's just looking at the heparan sulfates, so the post-translational modification of proteins. Typically, there is a sugar moiety that is on the extracellular surface, and you can stain for that. You can look for that. There is an assay that you can do, and that monitors if there is any sugar on the surface. And then there is a more precise way, which is using antibodies directed against the extracellular or intracellular domain of proteins. And you do it panel after panel. It's a very tedious process, and you got to do it extensively, at least for the main proteins you look after. And in these 2 papers, if you look at them, we really focus on 8 proteins. We isolate almost 200 proteins, but we know very well in detail the function and, the structure of 8 of them. And so for those, you actually measure responsiveness to an antibody. And if there is a better way, I'm all ears, because this is something that all reviewers regularly ask. So if there is a better way to do it, I'm eager to learn.
Thank you.
Thank you.